I wear the hockey jersey. But then when I go out of the office,
Oh, I see. I see. Makes you look more professorial. Although you need the patches on the elbows, really. Where are you going to be most of the time?
Probably. I tend to be a pacer, but probably just always.
Okay, just so I know where to point this. Lindsay, I'm going to come and perch next to you. I don't smell, I don't think. But it's not worse than usual. It's unfortunate that got caught on the scene. Yeah, a couple of minutes. I saw some people loitering around the most delicious sausages in the break room.
Yeah.
And donuts. So I tried buttering some of those.
Yeah, Di was telling me that there was food.
I had to close my office door just so I can smell most delicious sausages.
Oh, I don't. I can control myself around donuts. I'm not a donut person.
Yeah. Yeah. Absence is the best form of prevention in my case.
Yeah.
And just avoid the place. It's
I
better.
try to avoid sweets.
Hence avoiding.
Yeah, that's the secret, is to make sure that they never are. Yeah. Hey, how's it going? Oh, it's Friday, man, so that's something, right?
Yeah, that's your email. I'll do it. Yay, and Grace did spell that. Yeah.
Preparation.
They said.
All right. So,
I've actually put some in my bag and in my bag. Oh, what's there? All right, I also need a pen. You got a pen, Gabby?
You go.
Awesome. There you go. You can use it first. Thank you, Victoria. How's today's day? 27?
Yes.
There you go. You can get up side first. Robert, do you want an introduction or are you good enough to do that yourself?
I can.
Okay. Hi, I'm Robert. All right. It's a shame Sophie isn't here. She would love to hear about this stuff.
Salamanders.
Mm-hmm.
Sophie would ask hard questions.
Have you seen her latest rescue? Frilled crested gecko. That's it.
Oh, really?
Yeah. Called Tails because she lost her tail.
Sophie always asks hard questions, even when it's not about science. All right, so most of you know me. Those of you who don't, I'm Robert Page. I'm an associate professor of biology here at Texas A&M San Antonio. And this was just a really good opportunity to talk and meet with students. So thanks for the invitation, Dr. Crook, and basically thanks everybody for coming out. And so, like I said, a lot of you have some idea of who I am. Maybe everybody doesn't. So I'm going to kind of talk about a few different things, including kind of my background, what my training's in. It's pretty broad. I think unusually broad in some respects. I'll give a little bit of the history or background on the project, and then I'll talk about kind of what motivated this work. And so I'm going to kind of set a bad example, I think, in the sense that normally, like, I kind of stare down my nose at talks that aren't framed in a conceptual and theoretical framework. I tend not to like that. I was trained not to like that. I'm not going to make the effort to really do that today, though. This is because I like salamanders. I do like salamanders. That's mainly what the talk is about. But there'll be a little bit of conceptual stuff. But really what I'm going to be talking about is the past 5 or 6 years of research in my lab on plethodons. This is about a third to a quarter of what I do. I'll talk more about my interests in a second. But plethodon, these woodland salamanders, I'll talk briefly about kind of future directions. So I've gone to school at all different types of institutions. I did my undergraduate degree at a liberal arts college in Georgia. And so I'm really kind of a child of the liberal arts. I still believe in a well-rounded education. I don't think we offer that very much anymore, but that's to the detriment of society. That's kind of my personal opinion. I think that, and really when I was at Piedmont, I was studying vertebrate ecology. That was what I was interested in. I was interested in vertebrate natural history and vertebrate ecology. That's what I primarily studied.
I wasn't kidding.
Kind of thinking about, I wanted to work in evolutionary ecology as a master's student. I ended up at the University of Louisiana at Lafayette. I've actually been chased out of this swamp by an alligator. Believe it or not, it came up and gave that and ran me out. Got too close. Was a 5-foot female. I got a little too close to her nesting site. She let me know about it. But when I was at University of Louisiana at Lafayette, I was really studying behavior. So I was a behavior person. And kind of got a little disillusioned with that. So in between my master's and my PhD, I worked for a couple of years. And that's when I really started doing population genetics and conservation genetics. And so I kind of, it's like, okay, I've done sort of the ecology and evolution thing, and sort of the top 10 kind of thing, this is maybe kind of a middle ground. And I knew I wanted to go on and do like genetics and genomics. So I ended up at the University of Kentucky, where I really was doing developmental genomics. That's basically what I was doing. So really my PhD was not in population. My PhD was actually in developmental genomics and functional genomics. And then as a postdoc, I was really doing population conservation. Again, really kind of in between the master's and the PhD. So in terms of the type of work that I do now, well, obviously the population conservation genetic stuff is mainly what I'm going to talk about. But I still do, ecological and evolutionary development projects that I'm working on, including some projects that involve agent-based modeling to try to understand how different factors influence the evolution of basically life cycle variations. Working to write simulations that kind of provide insight into this. And kind of really the thing that sort of runs across all of my work, and in some ways probably more what I am than anything else at this point, statistical bioinformatics. So using tools from statistics, using tools from computer science and bioinformatics to really address questions in ecology, evolution, and developmental biology. That's how I would describe myself. So the project that I'm talking about, if I'm going to really sort of tell you where it's coming from, it's really coming from lizards. So this will probably make Watson happy. But about 7 or 8 years ago, my lab was doing an awful lot of population genetics on lizards. And there were these large lizards that are invasive in Florida. So things like Argentine black and white tegus, things like Nile monitors. And one of the things that we did to complete this work was a lot of sequencing to generate molecular markers. There weren't really any molecular markers for the type of work that we wanted to do in these species. And so we ended up doing a fair amount of sequencing and then kind of using informatics to identify markers and then use those markers to actually do the work. And one thing that you can do with high throughput sequencing is you can multiplex samples. And so I started thinking, we've got some lizard samples. We may have sequenced some spider stuff too back then. I thought, why not throw a salamander sample in there? I like salamanders. You never know when the salamander sequence might come in handy. So I didn't really, I was really just thinking kind of like buy one, get one free. That was about the logic, like we're going to pay all this money for sequencing. So why not? Hey, let's just throw a salamander sample in there and see what happens. That was really all I was thinking. So when we did all of this sequencing for these projects, really what we were focused on, I kind of snuck a salamander sample. We're trying to see the consequences of that later. Now I'm going to switch gears and talk a little bit about Plethodontidae. So Plethodontidae are a family of salamanders, actually the largest family that's still on the planet. Over 400 species, 25 genera. And what you're really looking at here is like a heat map of diversity for this group. So you can see they're pretty widely distributed. You can find them in both Eastern and Western North America, down into Mexico, Central America. There's a few forms that occur in South America, a few forms that occur in Europe, and a few forms that occur in Asia. Now, basically on this heat map, darker red means more diversity. And so maximal diversity for Plethodontidae salamanders is up and down the East Coast of the United States, and particularly in the Appalachian Mountain Range. And even then, particularly within the Southern Appalachians. So that's where I grew up, is the Southern Appalachian. And so when I was an undergrad, my undergraduate mentor was a salamander guy. He took me out and run around, chase salamanders. And I thought that, this beats like staring through a microscope and having my eyeballs pop out of my head. So that's why I thought, well, I can do salamanders. Now, the particular genus that I'm going to talk about, Plethodon, these are fully terrestrial. They're what we would refer to as direct developers. So it's not quite right to say that they don't metamorphose, because when you look at them in the egg sac, they have external gills, and you can kind of see the larval features. But by the time that they hatch, they've already gone through that process, and they emerge as little miniature adults in the forest floor. And so there's no free-swimming larval period in these organisms. They don't have any lungs, so they've got to maintain high moisture. They do almost all of their respiration across the skin. So they've got pretty specific requirements. High humidity, that sort of thing. Yes, sir.
Does that limit the size of salamanders, the ones without lungs, having to do gas exchange across their skin?
We'll have lots of answers to that. I don't think about those types of questions.
Well, the largest salamanders in existence right now are they breathe through the skin.
Oh, okay.
It probably has some limitation to terrestrial.
The biggest ones have lungs for the most part, but
But yeah, your surface to volume ratio, as you get larger, as you know, the volume is going to get smaller and the surface is larger. So it's probably
it's a limitation.
Huh.
That would be my hunch as well.
but big stream salamanders.
So basically, these things were kind of where I grew up. There's interesting biology there, and they can be really locally abundant. So in terms of getting sample size and that sort of thing, they're really good to work. Now, I like to put everything in kind of a phylogenetic context, because like I said, this is really just kind of my I like salamanders talk. So I'm not going to work that hard to try to frame it in these, kind of highfalutin sort of way. If we look at Plethodon, the genus Plethodon, you see is that there's 58 species, and that there's two major clades. So there's this western clade that's very small, basically kind of right here, and then everything else is the so-called eastern clade. Within that eastern clade, there's a number of different groups. For example, the Glucnosus. The group that I'm going to focus in on is this Cinereus group here. So there's the node that corresponds to the common ancestor of all 10 species in the Cinereus group. It basically starts at Electromorphus and then goes down to Serratus. So I'll kind of show you that a little more closely. There's 10 species in this Plethodon Cinereus. So there's kind of a blow up. This is actually from one of Heiss's papers instead of Lean's paper, but basically shows the same topology. We can see all 10 groups represented here. Now, what's kind of interesting about this is that when we go in and look at the geographic distributions of the 10 species in the Plethodon Cinereus group, we see a very, very wide range of sizes. So on one extreme is kind of the namesake of the group, Plethodon Cinereus. And so that's in gray here. And so it basically starts in like southern North, or I should say about central North Carolina. Goes all the way over past into Minnesota, up around Lake Superior and into Canada. They're a very broad range. And kind of one thing to understand here is that, about 10 to 15,000 years ago, everything was glaciated, right? Except for this kind of southern part of the range. So all of this expansion out of southern Appalachia has happened over the past, say, 10 to 15,000 years. And what's interesting about this, if you go and study these organisms in the field, the conclusion you're going to draw pretty quickly, whether you use ecological methods like capture and recapture, whether you use genetic methods like I do, is that these things don't move very much. So there's this kind of contradiction where clearly there's all this movement that's taking place. There's no other way it could be. But when you go and look at it, it doesn't look like they move very much. Now, kind of in the middle, we've got these sort of regional distributions that I would talk about. So things like Hoffmanized, and just to kind of give you some sense of scale, this is like Blacksburg, Virginia. And then up here's Pittsburgh. You can see DC there. So we're talking about kind of a regional distribution here. Similarly for Richmond I, here's Louisville, here's Charlotte. So you can kind of get a feel for how much area we're talking about there. And then Electromorphous is another one that's kind of similar. Here's kind of Louisville again, Cincinnati, Cleveland, Pittsburgh. So you kind of get a feel for that. And then there's something that I would describe as regional, but it's disjunct. So Serratus, you basically got kind of three patches. What's probably going on here is that this species used to be kind of contiguously distributed across this entire area, and that it's gotten isolated over time. Now, on the opposite extreme, we have the 5 mountaintop endemic species of the Pleptodon cinereus group. So Nettoni, Virginia, Shenandoah, Sharonda, and Huberti. And this is a 50-kilometer scale bar. So you can see just how small some of these distributions are. Indeed, they're amongst the smallest distributions of any vertebrates in mainland North America. And that immediately makes these things of conservation concern. And the reason for that is that if a fire rips through this habitat, or if a disease rips through this habitat, or some other type of disturbance, you could potentially be looking at extinction. The other thing that's going on is that all of these things have their ranges completely engulfed by this thing. But they're surrounded on all sides by what's clearly, and I'll talk about this more, but what's clearly sort of a ruderal, weedy type of species. So I'll come back to that. But I kind of want to set the stage for this, because really, after I kind of get through a little bit more preliminary stuff, where we're going to go is what's going on with the distributional dynamics of Pleptodon cinereus with respect to some of these mountaintop things. So in terms of the methodology, old school pop gen, right? We're moving towards, I'm not even sure I'd say new school pop gen at this point, but more modern approaches, right? So going to do genetics, you got to have tissue, right? So go out in nature and collect tissue. You're going to do genetics, you got to have DNA, isolate DNA. In this case, we just did a lot of shotgun sequencing. So if you're not familiar with shotgun sequencing, the idea is that you just generate a bunch of random fragments from the genome, and you don't worry very much about where it's coming from or how you sort of bias things or anything like that. I'll come back to this as well. But the idea is we're just generating random fragments from the genome. So then what we can do is we can go in and informatically scan and ask, okay, what's in those sequences? And what we're really looking for is microsatellite, sometimes we call SSRs for simple sequence machines. But the basic idea behind like a microsatellite is that you might have something like this. It's repeating over and over again. Or it could be a different motif. This would be what's called a dinucleotide microsatellite, AG. So 2 nucleotides repeating over and over again. Or you could have like a different motif. You could have a tetranucleotide motif, or you could even have, say, a pentanucleotide motif, something like that. And the reason that people tend to use these microsatellites for population genetics work is because they're hypervariable. And because of their repetitive nature, they're prone to undergoing something called translinkage, where the polymerase breathes and either backtracks or jumps ahead a repeat unit, and that results in either expansion or contraction of the repeat. And so basically, one way of thinking about variation in microsatellite design is how many repeat units are there, right? So here I've got 1 repeat, 2 repeats, 3 repeats, right? And of course, because these things literally do correspond to insertions or deletions, the length of the PCR fragments, if you just prime in the flanking regions on either side of the microsatellite, will differ in length, will differ in size. And so what that means is that you could genotype by sequencing, but you can also use electrophoretic methods to genotype. These are codominant markers where you can detect both alleles that are present at the same time. So scan for these FFRs, identify them informatically, then go into the lab and start saying, okay, which one of these things are really going to work, right? Because in molecular biology, where the rubber meets the road, is at the bench top. So once you've got your SSR loci you're going to use, basically you start your genotyping, you do all of your PCRs, you do all of your fragment analysis. You've got a fair amount of data processing you have to do. Then once you get your multi-locus genotypes, you start asking questions, right? So what's the variation like in my populations? Are there any meaningful population genetic parameters that I can estimate from my data, like the effective population size or gene flow rates, for example? And how much variation is there between populations that I might consider? So that's kind of the logic. And this is kind of a generalized methodology for everything I'm going to show you. So to kind of start at square one, basically marker development. So back in 2017, I guess it was, Alex Cameron, who was a master's student at John Carroll University, and Jeff Anderson, who was an undergrad in my lab. And I published a paper in PLOS1, in which we finally got around to describing that random salamander sample that I talked about, multiplexing with all of those lineages. And so we did a bunch of sequencing. We identified a bunch of fragments that contained SSRs. But what you're really interested in, because having a fragment that contains an SSR isn't very useful if you don't have enough left flanking region and right flanking region to prime into. Because if you can't amplify it, then PCR is not going to work. So you don't only need a fragment to contain an SSR, but it needs to have enough flanking region on either side of the SSR to be primary design. And so that's the distinction between the left and right-hand bar chart. When we had enough flanking region, we would refer to it as a potentially amplifiable locus or a PAL. So this is just fragments that contained SSRs. This is ones that we actually could design primers to and do some genotyping. And all of this stuff at the bottom is just dinucleotide, repeat motive, trinucleotide, tetranucleotide, pentanucleotide, and so forth. So we discovered, a bunch of markers, and part of the motivation for doing this is because these animals don't move very much and there's marked geographic differentiation. Markers discovered in one region of this species won't necessarily work very reliably in other regions. And the reason for that is mutations to the flanking region. Now, we wanted markers that would work up in northern Ohio, where some of my collaborators were working, and markers that would work down in southern Appalachia, where we also. So we screened a bunch of them, identified a couple of dozen loci that fit the bill, and then just really kind of did a bunch of blue collar stuff to demonstrate that it works. So people would expect isolation by distance. This is a measure of genetic differentiation. This is a measure of distance. Looks like isolation by distance to me. People would expect for you to be able to separate out different regions, populations in different regions. This is just a multivariate approach based on multi-locus genotype data. It's called discriminant analysis of principal components. Doesn't really matter what it is. But the point is we can separate out southern Appalachia an isolated island population in the middle of Lake, what lake is that? Lake Erie for Cleveland. And then this is like basically all of the mainland sort of Ohio stuff and kind of Western Pennsylvania. So we get that regional separation that one would expect. It's just kind of proof of concept. Here's a bunch of markers and they work, right? So kind of some resource development. Now, I remember going to a meeting around this time and just kind of telling people like, hey, I've got this resource that my lab developed, and would anybody, like access to it, maybe to collaborate, use these markers to do stuff. And there were a number of positive responses to that. And one of the positive responses came from Sean Kuchta and his graduate student, Maggie Hantak, and some of you may remember, Maggie was here, gave a talk actually in this seminar series about a year and a half ago. And Maggie was as hurdle working a graduate student as I've ever seen. She had, let me look at the numbers just to make sure I get it right. And she collected 648 samples of individuals across 28 populations for about 23 individuals per population across these 28 populations, all up and down the kind of southern shore of Lake Superior or Lake Erie in northern Ohio. And what Maggie was interested in was what evolutionary forces maintain color polymorphism. It turns out that this species has a color polymorphism. There's the so-called leadback salamander, which is shown here. It doesn't have a little stripe. And then there's the so-called redback salamander that has a stripe. But they're the same species, Plethodon cinereus, and we can find them in the same population. It's just a color polymorphism. And so what this is showing you is like the frequencies of the color morphs across Maggie's sampling range. And you can see that if you kind of stare at it just right, there's a little bit of a climb there with more leadback on, in the west and more redback in the east. And so I don't want to go through Maggie's work in detail because she'd explain it much better than I would, but it is a lesson for graduate students. And since this talk is primarily for graduate students, I'll kind of point a couple of things out. We hit these data with virtually every analysis you could think of. So we constructed surfaces in GIS like canopy cover, ecological niche model, waterways, digital elevation models. Used all of those things in regression style models with they're basically maximum likelihood with population effects models. And, a little bit of stuff would come out, but we really tortured these data. And basically, there wasn't any clear story. There wasn't any clear pattern. And so, here's Maggie. She's done all of this work. And it's good work. And she's been incredibly rigorous in what she's done. But there's just no clear story. You can kind of see the way I've summarized it. Well, it's complicated. There's probably, different evolutionary forces that play a role. Sometimes, though, if you do a good job and you work really hard, and there's no doubt about the integrity of the data and the integrity of the analysis, even if it's not necessarily the clear story, you can get it in a good journal. Ecography is a good journal. So, would I encourage you to do high-risk projects like this as a master's student? Probably not. But if you are, do a really good job. You never know what might happen. Right? Now, another person who responded to my kind of call for anybody want to work with me? I've got a bunch of markers. Was an old friend of mine, Carl Anthony, and his wife Carrie Ann. We kind of run a lab together at John Carroll and their student Alex Cameron, who was actually kind of part of the initial description paper as well. He used to be an author. And what Alex was interested in was kind of the age-old question. I mean, in some sense, what Alex wanted to do was something that's been done a bunch of times. But what's the effects of urbanization on gene flow? relative to like contiguous habitat. And so he had basically sites in the Cleveland area. And then he also had sites in Allegheny National Forest, kind of maybe 80 miles east of there in western Pennsylvania. And he basically set up his sites where in the two different landscapes, the spatial arrangement is almost identical. So he's kind of controlling for distance in the way that he's arranged it. And what you're looking at here is the Cleveland area. Basically, red is like urbanized, like city habitat. This is habitat suitability. Blue means better, red means worse. And then this is the forest habitat. So this is all just like tree cover. And then still blue means better, red means worse. And so you can see there's a huge difference in the way that these landscapes are set up. And so what you would kind of expect, and indeed what most people kind of see when they do this, is that there's quite a bit of genetic structuring within the urban habitat, but less in the contiguous habitat.
Robert, can I ask you, it's probably a silly question. How do you, because I don't know this, how do you calculate habitat suitability and what's that mean?
Habitat, so it's going to be based on your intuition, but you do it within GIS, you would basically specify within each raster, you know, what your kind of intuition of habitat suitability is. And that was actually validated. So Alex and James Watling, who was actually the GIS guru on this. So I do a little bit of this. Great.
So is that habitat for that species of?
Yeah,
Gotcha.
you got it. Yeah, so and James and Alex actually validated that model so that they would like construct their measures and then go out into the field and see if they would see greater abundances where they and they did. I just don't have space to kind of cover everything.
Cool, okay,
Yeah.
cool.
So
Cool. I need you to train me on that.
So yeah, so we did see different structure, right? So if we go and look at the urban sites, we see that there's this one population AR that's really different from the others, and the other three are, you know, pretty homogeneous. And then when we look at the rural sites, we see kind of basically they're all part of the same genetic group. And if you're not used to these kind of plots here, what these plots are bar plots. So I'll try to draw one up. But basically for each individual, there's a bar, right? And each genetic group that you've identified just purely using the data gets a different color or a different pattern. And so if there was an individual like this, let's say, all right, and let's just say that this is, you know, 0.2 of that bar, and this is 0.8. Well, what that implies is that 80% of this individual's genome is derived from the no pattern group, and 20% is derived from the pattern group. So pretty much the same thing going on in the contiguous habitat across sites, but there's one site that's really different in the urban habitat. So, you know, we went in and did a bunch of stuff, but really what it boiled down to is kind of shown here. So what we're looking at here is theta is basically an estimate of effective population size. So if you're not familiar with this, theta is for any mu, where mu is the mutation rate, n is the effective population size. And so basically larger values, we're generally kind of assuming that the mutation rates are pretty much the same across populations. So it's basically related to the effective population size. What we can see is the site that's really different has a much smaller effective population size. So drift is stronger and is pulling it apart. And gene flow, which is kind of what's shown with these arrows, asymmetrical gene flow and gene flow rate, isn't really enough to stop that. On the other hand, when we look at the gene flow rates in the contiguous habitat, which is over here, and in the urban habitat, which is over here, they're all about the same magnitude. And really, this is all seems to kind of be driven just by strong drift at this one side. And so the conclusion is that because this is such a rural kind of weedy species, that it can actually maintain levels of connectivity across this urban environment that are more or less consistent with what we see in the contiguous environment. And that's probably part of why Cinereus has so much bigger range than the other members of the group. It's tolerant of marginal habitat. It's a ruderal species. Now, at the same time that we were kind of finishing up this work, my buddy David Marsh, you may also remember, he was here about a year ago, last spring, asked me, would you like to do some work on some of these mountaintop endemics? I was like, yeah, yeah, I'd like to. And so I got some students here. Some of you have been here for a little while, may remember some of these students. But we started screening some of these markers that we developed to see if they would cross amplify in Plethodon huberti, which is this very narrowly restricted species. And so we basically screened these markers to do the work so that we could do some hybrids. Because what David had been seeing when he was working in the field were animals that looked kind of weird, maybe somewhat intermediate between Cinereus and huberti, or at least a little bit off in their coloration patterns and that sort of thing. And so we started thinking, I wonder if these things are hybridized. And so we went into the huberti range. So this is kind of Cinereus's range, sort of a blow up of this little region here. There's huberti's range. Notice that the scale bar here is only 100 kilometers, so that's tiny. And then this is like a big blow up. The scale bar here is about 3 kilometers. So we're talking about a species that's found along about a 20-kilometer stretch of ridgeline in the southern Blue Ridge Mountains and nowhere else on the planet, right? And it's very mountainous, so you can kind of see the contour lines. So we have a site that is pure huberti, a reference site that's pure Cinereus, and then we had kind of sites in the contact zone where both are present. And so the initial question was, are these things hybridized? Because things like genetic swamping or genomic swamping can lead to extinction. If you've got a situation like this, where you've got this kind of narrowly restricted endemic that's surrounded by this really common species, and they come in and they start hybridizing, well, who do you think we're going to back cross into more often? And eventually you just kind of hybridize yourself right out of existence. And so that was kind of the concern there. So, kind of set this up. So Cinereus huberti, that's self-explanatory. PHCZ, that's huberti that were collected from the contact zone. Floyd's Field, that's our huberti reference site. Cinereus collected from the contact zone. PHRID is Thunder Ridge. That was our Cinereus reference site. So, you know, there's a little bit of noise here, and I will mention that the asterisks correspond to the animals that David saw that he thought looked a little different. So those were the ones where we were really kind of looking to see, you know, maybe there's something going on. But there's not. You're going to get a little bit of noise in these values just because there's an estimation process, but there's nothing very convincing about this. It looks like two pretty well separated groups. This is a more explicit model. It's implemented in the package New Hybrids. And what New Hybrids will do is it will actually try to classify individuals into different hybrid classes. So it will classify them into pure Cinereus, pure huberti, F1 hybrids between the two, F2 hybrids between the two, or F1's back cross to Cinereus or F1's back cross to huberti. And so there's different colors for all of those categories. They just don't all turn up in the graph because they're really well separated. We were smart enough to make the redback salamander red and to make huberti blue, right? So there's an intuition. So there's this one animal here. And in these different plots, this is a Bayesian procedure. So there's different priors. In Bayesian analysis, that's what drives the frequentists crazy about Bayesian statistics. The priors. You can load them up to make it say whatever you want if you're not an honest person, right? But at any rate, depending on how we set the priors, yeah, maybe there's one hybrid in the whole sample, but even that's equivocal. Certainly no strong evidence of hybridization. This is an AMOVA just to show that they're really well separated, right? So you're always going to see more within species are kind of more within group variation than between group variation, kind of overall. So 41, almost 42% of the variation in the data associated with differences among species, but that's actually really high. And you look at like the FSD estimates and all that stuff, these things are separated out. And just to kind of beat the dead horse, most various statistics that aren't model based, so the same thing. You can separate them out, it's not really local. So we answered that question. It doesn't look like hybridization is increasing the extinction risk of Plethodon cubica. But when we just kind of looked at variation, there was a pattern that we didn't expect. Common sense kind of tells you that you're going to see more genetic variation in the broadly distributed, very common species than the narrowly distributed, kind of rare species. But that's not what we saw. So this is a haplotype network of cytochrome B for Plethodon cubica. So you can see there's a few different haplotypes. When we looked at Cenarius, there was only one. These are the nuclear data that we generated on microsats. And this is kind of a complicated table. It's what's called a hierarchical diversity petition. So all of these diversity measures are scaled to vary between 0 and 1. 1's basically like maximal diversity and 0 is basically like none. And so we're kind of starting with among species, within species, among populations within species, within populations within species, and then among individuals within population, and within individuals, which of course is heterozygosity, right? And so it doesn't matter where in the hierarchy you look. And there's statistical tests in parentheses. Doesn't matter where in the hierarchy you look. Cubica is always more diverse than Cenarius. Kind of the opposite of what you would expect. So that was a little bit of a head scratcher, but we got to thinking, well, if they're more diverse, maybe they're also more differentiated. And so we went in and did just some kind of basic top gen on the Cubica samples individually in the Cenarius. So we kind of separated it out by species and species analyses. And when we did that for Cenarius, basically there's no structure across our sites. We go in and try to estimate how many genetic groups are. The highest one, the one that has the highest likelihood is just 1. And then when we did that for Cubica, the highest likelihood was on 2. And so when we kind of go in and look at Cubica, that's what we see. So there's certainly gene flow between the contact zone and the reference site, but some structure. So one thing that could be driving this pattern is, I mean, we know that Cubica is a mountaintop here, and Watson could probably tell you more about this than me, but one of Joe Bernardo's ideas actually was, you know, they're gonna have perhaps an adaptation that involves lower metabolism because of the high altitude, and that could be associated with less movement. So maybe what's explaining this is differences in movement ability. Now we estimated that genetically, and we didn't really see much going on. But David's an ecologist, you know, and so he likes to do like these ecological things. And I'm just always like, that's just keep the samples coming, you know. And so David did this kind of contrived experiment, but basically what he did was take advantage of the fact that this thing shows site tenacity in the home. And so you can take elastomer dyes and put them in a syringe and inject them under the skin of these salamanders and mark them that way. So that you can go, you know, so it might be something like this. Where you pick up a salamander and it's like blue, green, yellow, red. You're like, oh, your salamander, blue, green, yellow, red. I'm going to do it, right? Or it could just be, I'm going to mark it, you know, blue for control, red for, you know, my 15-meter treatment or whatever it is, 15 meters, and yellow for my 30-meter treatment. So you mark a bunch of them. Some of them you basically put them right back where you captured them. Some of them you displace a random direction 15 meters away, and others a random direction 30 meters away. And then you just go back out there and you start seeing what you can capture. You're going to recapture the same individuals, and that tells you something about movement ability if you do it with both species. So I'll come back to why this is contrived in a minute. But basically what we saw when we did this, by we, I mean David, is that we would recapture about, a third of the controls. Okay. And then for both the 15 and 30 meter treatments, recaptures were about 2/3 of what they were in controls. Right? No difference between species. And like a logistic regression sort of model. Now, I know that you guys are good ecologists to know that homing behavior and dispersal are not the same. Right? Dispersal oftentimes takes place at a different life history stage than homing would, may be controlled by different genetic architectures and different circuitries. So, not really what we're after here, but still at least a measure of the ability of these two species to move through the matrix in which they find themselves. Right? I like the genetic data better, but we actually had to fight to get this in the paper. They wanted to relegate it to supplemental materials, but their policy was we don't like supplemental materials, and so I was able to argue, because David really wanted it in, I was able to argue, you guys say you don't want supplemental materials, and then we just include it, and they're like, yeah. All right, so this kind of got, we're sitting there kind of going, okay, I think maybe I see what's going on here. What's probably happening, because if you look at who's Hubert guy's closest taxon, right? Its closest species. Well, it's Medina, which is another one of these range-restricted mountaintop endemics that's not very far away. And so these things were almost certainly part of, some kind of common ancestral species that was more widely distributed at one point. That's kind of part one. The other thing to kind of be aware of is that even though there's this tiny little range here, Huberti can be locally abundant. It can reach levels that are as high as Cinereus. And just to kind of blow your mind, Cinereus can reach levels of, what were Alicia's numbers? Something like at Mountain Lake, something like 2.8 animals per meter squared. There's more biomass tied up in Plethodon cinereus in eastern forests than probably all other vertebrates combined. Okay, so we see what's going on here. We think Huberti has been evolving in this region in situ for a long time. That means lots of opportunity for mutation to introduce new variants. The local population sizes are high, so drift is weak and not eliminating variation quickly. And so you end up with a lot of variation in Huberti. What's probably happening, and I'll talk more about this in a second, is that Cinereus is a relatively recent invasion into the range of Huberti. And so if you're a relatively recent invader, that suggests that you've probably undergone a founders. And there's a reduction in genetic variation associated with that. And indeed, the levels of diversity in Cinereus, like microsatellite diversity that we see around Huberti, is lower than what we see in other parts of southern Appalachia. So one way of kind of trying to test this idea is using something called approximate Bayesian computation, or ABC. It doesn't matter too much what that is, but I'll give just kind of a brief conceptual kind of explanation. I can dream up different scenarios of how things have happened in the past, both from a demographic and an evolutionary perspective. And I can then carry out simulations, typically using coalescent simulators. So kind of reverse time simulators. To basically generate the patterns of genetic diversity that you would expect to see under the different scenarios that you think are plausible. And then the scenario that generates the simulated data that most closely match your actual data, that's the one that there's the most support for. That's kind of the logic here. Another nice thing about ABC is that you can estimate meaningful parameters, so effective population sizes, and also things like how far back in time splits occurred, or how far back in time admixture occurred in some cases. So what we see when we just kind of specify the really, really simple model, because remember, we weren't doing this study with this in mind, we just discovered it was really about hybridization. So this wasn't really set up to do this, but after we saw the patterns, we thought, well, let's see what's going on. What we see is that they both, all four of the populations we were looking at, we just kind of pulled the context of. But all four of the populations we're looking at have pretty big effective population sizes, and they're actually nested kind of within this sort of hierarchy of populations where it's kind of hard to say where a population ends because it's just kilometers squared and kilometers squared of contiguous habitat out in the national forest out there. But what we see is that the split between the Plethodon huberti reference site and the Plethodon huberti contact zone, the best estimate on that is that occurred about 585 generations ago. And the best estimate for the split on Plethodon cinereus contact zone and reference site is about 90 generations back. So that just kind of lends more credence to this idea that what we're really seeing is this accumulation of genetic diversity because huberti has been evolving here in situ for so long. It's maintained large effective population sizes and drift is weak, and cinereus is a more recent invader into the area. Now, conservation consequences. Basically, back in 2007, I think it was Dean Adams at Iowa State, wherever he was. And they constructed a niche model for huberti. Basically, they concluded that cinereus could use all of the huberti range, and really the only thing that's keeping it out is huberti. Right? And so the concern is that climate change is starting to change what's going on the mountaintops in a way that perhaps makes it less suitable for huberti and more suitable for cinereus. And so as cinereus encroaches on huberti, there's this concern that this basically interspecific competition that huberti has been able to kind of hold at bay is going to be bad for huberti. I mean, under the worst case scenario, it's the beginning of the end, right? So normally we kind of say, well, it's not that big of a deal. They're going to have some time because they can track up the mountain, right? So like if you're an animal and the climate's changing, it's warming. So that means that, you can't really see it that well, but let's say, you're here, climate warms, maybe you go up to here and you actually track the climate. So you kind of maintain what you're used to. Problem is if you're a mountaintop salamander, you're already at the top. So where are you going to go? And these things don't move far enough to like go all the way down a mountain and up some other higher mountain. So it's potentially a pretty serious situation. We're arguing that this is something that should be incorporated into the management plan for huberti. I know I'm almost out of time, so I'll basically not really worry about ongoing work and just kind of go ahead and shut things down. But before I do that, I do want to acknowledge the people who contributed to this. So certainly there's a number of undergrads from my lab, listed there, and also David's lab, graduate students from Carl and Carrie's lab and Sean's lab. And of course, Sean, Carl, Carrie, James, David, some of the other folks at WMU like Kelly, our funders, particularly Fish and Wildlife Service, who gave us a little money to do what we're working on now. So with that, I'm happy to take any questions.
You got any questions?
One thing you can learn from the point is that a good story. Well, Dr. Shade certainly students of people can express your science in a story like he had. It's going to be much more interest, and also develop network because you took him 5 minutes at the end to talk about all the people that have developed his network. So more of a comment there, but I think you can learn a lot about science from things that aren't necessarily part of the nuts and bolts presentation.
Yeah.
I was gonna keep that city question. So when you're doing that 15, whatever it was, meters, 15 miles?
Rehoming one.
Oh, 15 meters.
Meters?
Yeah. That's a long way for.
No, I know.
so it's not-
Did you ever do meters?
It was a random direction. So basically you kind of think about 360 degrees. I mean, you know, like an R, I don't know if you do R, but if you do R, it would be like 360, and then it just be basically a sample of one to 360. And then we just define whatever, you know, 0 is, and then hit we're in and draw.
There is no way to account for this kind of model that you're talking about, it's going over, going over this.
Oh, I see what you're saying. Yeah. I think that, I don't know exactly where David did that experiment, and so what the elevational component there is. But he's not really talking about this in the point. I think he's talking about sort of just kind of equivalent space.
So this is one of what, 5 mountaintop endemic in that area?
Species, there are many others in some of the other counties.
Has anyone looked at any of the other ones for potential hybridization or encroachment?
In the glutinosis group, a ton of that work's been done. There's kind of a long history of that. And in that group, you oftentimes see it. There's all of these different slimy salamanders that basically all look the same. You can't tell whether any of them are and sequence them or you know where you are and where you found it. And you can just use the ingredients to say. But in the slimy salamander group, hybridization's pretty common. It seems like in Cinereus less so. I know that Rick, what's his name, up at College of Worcester, at one point did some stuff with Electromorphus and Cinereus, and you know, said that they're hybridizing, but he's an ecologist and he doesn't know what he's doing. He collected like across like 2 genes and identified 20 steps. And they had 20 markers. All through. Now I have 2 markers. And then, you know, Sean then went back and pointed out, he was nice about it. He went back and corrected it and, you know, published a correction. But it looks like in Electromorphus and Cinereus, it's not happening. So in the Cinereus group, it seems like it's less common.
So what are the barriers? Oh, sorry.
No, you go.
What would the barriers to that be? I mean, they're not geographical, clearly, if they're able to live in the same area.
Yeah, what would the barriers be? So I think it's really, there's a lot of different things that could happen. But what Dean Adams' work suggested is really the main barrier is Huberti. Huberti just simply already being. So it could just be an order of occurrence kind of, right? So I mean, like that would be the simplest model. I'm not saying that's what it is, but I mean, imagine, you know, you've got this thing here, you've got this thing. You've got that thing there, right? And this thing's trying to get in. But let's say that like already being somewhere is an advantage when it comes to the competition. Well, you know, it basically can block that simply because it's already there. On the other hand, if you're at the top of the mountain, that environment's starting to decay with respect to this. So now this thing's got a competitive disadvantage that it didn't used to have. It starts to weaken the defense. I'm not saying that's what's going on. It could be way more complicated than that. But I'd see that as kind of a null case, right?
Along those lines, are densities stable as you go up the mountain? The species or
For
in and out as you go?
For Huberti, the transition's gradual. So what you'll see is it's like kind of scenarios kind of here, and then kind of Huberti here. And what you'll kind of get is something, I guess I'll sort of draw it. Kind of like this. Right, so we've got kind of a bunch of scenarios back here, and as you kind of go in, it sort of starts to taper, and then it eventually gives rise to kind of Huberti on the other side. But for Sherrado, which is one of the other ones that we're starting to work on, that's not the case. It's like a really, really abrupt transition. And that's part of why we need to look at more than just one species and really kind of see if is there any generality, or do you really need kind of just all sort of to conceptualize completely distinct management plans for all of these things, right?
Just quick, for the reverse time simulations that you compared your actual data set to, how large did they have to?
Oh, so what did we have? It's been a little while since I'd have to look at the tape. I want to say that we probably had, yeah, I think we had a few more scenarios than Hubert Guy. We probably had in the neighborhood of, like 100 scenarios and maybe 75 Hubert Guy or something like that across all the sites.
I have some questions about topology in general, and I don't know if you can answer or not, but I'm thinking about speciation and taxonomy here, and how would you generalize a location where a speciation where that split occurred, and in that location where these are now mountaintop dwellers, could they have once been, down in the valleys where it was generally different, and that's, as they started moving up the mountain, that's where there's separation.
Yeah, indeed. And so let me pull up the map. Give a kind of visual reference. But yeah, the short answer is yes. So if we
How do you determine a point of origination for, one of these species based on ACV or that split that happened so many years ago and stuff like that? Or,
yeah, so that's like a phylogeography.
okay.
Yeah, so you can ask. She'll tell you more than I do. So yeah, I was kind of arguing that these things are closely related and probably used to be part of something that's contiguously distributed, right? Probably what happened there is that it warmed at some point in the past.
Sure.
And as it warmed, the lower lying areas in between basically became less suitable. And so what was once something that was distributed across both low and high, kind of high altitude areas, basically got isolated to the mountaintops. That's what we think happened.
So what factor of natural disaster or, human interaction?
Well, this would have been far enough in the past where humans wouldn't have had much to do with it. But I mean, sometimes things just change naturally, right? So if you think about like 64 million years ago when the dinosaurs were wiped out, I mean, that was kind of a cataclysmic thing, but there wasn't really anything unnatural about it.
No.
And I kind of argue the same thing actually with humans. Like there's not, I mean, like we're part of, the idea that like we're unnatural, I'm kind of uncomfortable with that. Or even that like, plastic or whatever is unnatural.
So
I think we better wrap up because I have a feeling someone's dying to.
Yeah. Sure, we're done. Thank you, everybody.